This video is all about meiosis, and I'm going to start by talking about what is really the end goal of meiosis. Well, we know that mitosis with a T is used to make new body cells, which are going to be our somatic cells. And the truth is, honestly, 99% of our cells divide mitotically, because most of our cells are our body cells. Your body is what you are and who you are. So what is going to happen with meiosis? It's a special type of cell division that is going to not make our body cells, but our reproductive cells called our gametes. Remember, gametes are going to be sperm in males and eggs in females. So another thing I want to mention before I move on is that somatic cells and cells that mitotically divide are what's called diploid, meaning that they have 2 of every chromosome. A lot of times it's written 2N, N being the number of chromosomes. Meiosis, on the other hand, makes haploid cells, which have half the number of chromosomes and really only 1 of each. So instead of having chromosomes in pairs, like our diploid mitotic somatic cells, meiosis is going to make cells that are haploid. And sometimes I say it's halfloid, which is just going to be N. And it's 1 of every chromosome. And another difference and comparison between mitosis, which makes most of our body cells, is that mitosis, all the cells created should be identical unless there's been a mutation. Meiosis, all of your sperm, egg, whatever, are supposed to be different from one another because you shuffle the genes. And I'll get into how that works later. So how does meiosis actually begin? Well, as with any cell division, meiosis is going to begin with interphase. Interphase comes before both mitosis and meiosis, and it involves 3 main steps. One of the most important phases of interphase is going to be the S phase, because that is when the DNA actually is synthesized and copies itself. So remember, our cells are normally diploid, meaning there's 2 of every chromosome. So what I have here are a pair of, let's say, chromosome lucky number 7. So why we give both of these chromosomes the same number is that they have genes for the same traits at the same spot. For instance, this gene here at the top would code for hair color in both chromosome 7s. This gene here might code for how your heart develops. And these clustered down here may code for skin color or something like that. But the point is, these genes, they code for the same traits. However, because these chromosomes come from different parents, one comes from the paternal side or father's side, and the other comes from the mom's side. And that's why you contain 50% of your genes from your dad and 50% of your genes come from your mom, because 1 out of every 2 chromosomes you have belong to one parent or the other. So because these chromosomes are not from the same parent, they're actually not going to be identical. So say both of my parents have brown eyes, which they do. That means that if this gene here codes for color of eyes, both of my parents could have potentially given me the brown-eyed gene because I have brown eyes. So those might be the same. And on the other hand, perhaps my dad has brown hair, and that's on this gene down here. And maybe my mom has red hair, and that's on this gene. In this case, they're not the same. My parents did not give me the same version of that gene or that trait. So for hair color, you see a difference on these chromosomes. The different versions for genes and traits are called alleles. Because these two chromosomes are the same genes in the same place, they are called homologous because they code for the same traits, color of eyes here, color of hair here, but they are not identical because your parents should not have identical DNA. Obviously, if you look at your parents, they probably do look slightly different, I would hope. They're not identical twins. It's not possible. So they have differences. That means their DNA is different. So what happens in the S phase is every single chromosome is going to duplicate. So we're going to duplicate this chromosome 7 as well as this chromosome 7. This in the end is going to give us a grand total of 4 chromosome 7s. And this is just one example of a chromosome. We as human beings have 22 pairs of autosomes, 1 through 22, and we have 1 pair of sex chromosomes, which are XX if you're female and XY if you're male. And in S phase, every single one gets replicated. And that means that in the end, we're going to have all of these pairs of 4. You'll have 4 chromosome 7, 4 chromosome 6, 4 chromosome 10. If you're female, you'll have 4 X's. If you're male, you'll have 2 X's and 2 Y's. So there's no chromosome left behind in this instance. So this is what we're left with after the S phase. We've got dad's chromosome here has been duplicated, so that's why these two are identical. And mom's chromosome here has also been duplicated, which is why these are identical. And we call these identical pairs sister chromatids. All 7 together are homologous because these two are not identical to these two. However, all the traits match up and will code for the same genes, but may have different versions of those genes, right? So different alleles for those traits, but the same traits in the same exact spot on the chromosome. And the reason why that's significant is it allows something called crossing over to be possible. So in prophase, all of these are going to connect to each other and trade genes, but I'm going to get to that in a second. So just to recap, it's important to note that this is what the cell actually looks like in S phase. Nucleus is still there. All that's happening is that our DNA is duplicating inside of the nucleus. And this is happening not just for one chromosome, but all 23 pairs of chromosomes that we have. So if we start with 2n, number of chromosomes, or diploid, which we have 23. So we normally have 46 chromosomes, but in this case, we are actually duplicating every single one of these 46 chromosomes. So in the end of the S phase, we will have 92 chromosomes total as we move into prophase 1. Prophase 1 is where one of the major events of meiosis occurs. And that event is called the crossing over. So if we zoom in on our chromosomes in the nucleus, all of these here are the same type of autosome. So remember, all of our chromosomes are numbered. So these would all 4, each line being a different chromosome, they would all be chromosome 7, just like the example I gave earlier. So this entire quad would be chromosome 7. And we call the quad a tetrad because it has 4 chromosomes all smushed together. And you can see here, they're kind of crossing over, which is what this is called, and they're mixing their genes up. Same thing with this particular chromosome pair down here as well. This would just be another set of chromosomes, so maybe chromosome 11. So all of these would be chromosome 11. And this again is a tetrad because it's 1, 2, 3, 4 chromosomes all combined. These down here, these green ones, would be maybe mom's chromosome that's been copied, and these here would be dad's chromosomes that have been copied. So remember in the S phase, this would be copied, so that's why we have 2 purples, and this green one would also be copied, which is why we have 2 greens, 2 per parent, and they're all going to find each other. All the chromosome 11s, all the chromosome 7s, all the chromosome 1s, 2s, 3s, 4s, they're all going to come together and form these tetrads, which we call the crossing over. The whole point of this, and the reason why the crossing over is such a big deal, is because it gives human beings a lot of our genetic variation. It ensures that you don't just look like one parent, but a combination of both. So it gives you the opportunity to inherit a decent amount of good genes from both parents that hopefully will outweigh any bad genes you may have received. So it kind of evens things out, and it's also why we don't look identical to our siblings, okay? And that's why even fraternal twins don't look alike sometimes. The only way that you're going to have any sort of identical DNA to another person is if you have an identical twin, and even then there are still some differences. So let's move on to the next phase of meiosis, okay? And remember, this is meiosis 1, dealing with homologous chromosomes, doing the thing. Nucleus is disappearing, so we're not going to see that in metaphase. So in metaphase 1, what's going to happen is that these homologous chromosome tetrads are going to move to the middle of the cell. So you can see they're still in the tetrad. So homologous chromosomes are going to move to the center, almost like they're all smushed together still. So they have not separated from each other yet. They're still mixing their genes, they're still shuffling everything, and they're still connected. This is going to line everything up, so when the separation happens, everything winds up going where it's supposed to. So let's take a look at the next step. So here's where things get interesting. So you can see that the tetrads, or the quads, have been broken up. So what we have now is a pair of chromosomes on either side. So these were all one tetrad, and now they're separated. So we have 2 chromosomes per X. So if you look closely at our X's, you can see that they are a combination of both original chromosomes. So when they replicated in the S phase, both chromosomes, mom and dad's, when they duplicated, were identical. So mom's was identical to mom's copy, and dad's was identical to dad's copy. 100% mom, 100% dad. However, because of the crossing over, now no chromosome is 100% mom or dad. They're all going to be this weird mixture of mom's traits and dad's traits. For instance, if my mom has red hair and blue eyes, maybe she traded here in the middle her blue eyes for my dad's brown eyes and kept the red hair, and he kept the brown hair. Now this chromosome has red hair, brown eye gene, and this chromosome here has the brown-haired, blue-eyed gene. So you can see they kind of traded some traits around, and that's happening on 1,000 times over. We are very complicated human beings, and we have a ton of traits that are all being shuffled. So I'm very simplifying this just down to hair color, eye color, but just know that there are thousands of traits that I could use as an example. So you can see that happened on all of them. And you could also observe that these are different than these, and vice versa with these down here. None of these X's are identical to the other, and that is going to contribute to the amazing genetic diversity that we see amongst human beings, which is why I don't look exactly like my brother, and you, if you have a sibling, you might not look exactly like them either. As a matter of fact, unless you have an identical twin, you don't look the same as your sibling, and it's because of the crossing over. In telophase 1, you can see these sister chromatids now, because it's just 2 of them, are still mixed up, still a hodgepodge of mom and dad genes, and the nucleus is starting to reform. That's about all that's happening in this one. It's that the cell's kind of getting ready to divide. In cytokinesis, the cell is finally split in two. We have one cell here, two cells here. They've got their OG, now kind of brand new unique DNA, and the nucleus is completely reformed. So both of these cells are going to have two of every chromosome, although now they're kind of not identical anymore. And they're also going to have 46 chromosomes apiece. So this kind of poses a problem, because if sperm and egg had 46 chromosomes apiece, that would not lead to good things when fertilization occurred, because then a 46 chromosome sperm would fertilize a 46 chromosome egg, which would lead to a 92 chromosome person. Not exactly what the goal is. So what we need to do is we need to divide one more time. So all that's really going to happen in prophase 2 is that the nucleus starts to disappear and the cell gets ready for division again. It's honestly a pretty uneventful step. The reason why we have to get rid of the nucleus is because it would get in the way of the chromosomes moving to the center of the cell, which is actually the next step. So in metaphase 2, in both cells we just created in meiosis 1, we're going to see the sister chromatids line up in the center of the cell. So these stages are actually very similar to mitosis. In mitosis, you'll notice if you look back at those notes, everything is talking about sister chromatids. So in mitosis, it just happens a single time where the sister chromatids line up in the middle, they separate, the cell splits. So in this case, meiosis 2 and mitosis, in terms of the way the steps are written, are pretty much the exact same. So in metaphase and meiosis 2, it's all about the sister chromatids lining up. And the next step is anaphase 2, where the sister chromatids are going to separate. And again, we have two cells because we made two cells already in meiosis 1. And you can see now one chromosome per pair. So remember, these two were just connected as sister chromatids. So now one chromosome per sister chromatid is going to head towards one end of the cell or the other. So you can see that now all we have are these single chromosomes, and they're all kind of different. Telophase 2, cleavage furrow forms, cells getting ready for division for real. Nucleus is starting to reform. Not really a lot to note about telophase 2. And last but not least, we have cytokinesis, where in the end, we are going to get 4 unique haploid gametes, each with 23 chromosomes if we're looking at a human being. So instead of containing 23 pairs of chromosomes like our somatic cells, heart, brain, liver, all have 23 pairs, your sperm and egg only have 23, and that's it. There is no pairs. So each one has 23 chromosomes total. And that is going to make a lot of sense because when sperm fertilizes an egg, 23 in the sperm plus 23 in the egg is going to equal a 46 chromosome person. And if you look closely, none of these particular egg or sperm, whatever you want it to be, is the exact same in terms of how the genes actually got distributed. So none of these are going to be identical, which is why I wrote 4 non-identical gametes. And they're not identical because of the shuffling of the genes and the crossing over that all started back in prophase 1 in meiosis 1. So that in a nutshell is meiosis and how it works and operates. And we'll get more into how this can go wrong in a different video and look at karyotypes and how you can tell if something has happened in meiosis that shouldn't have.